Okay, is it visible now to everyone?
Yep, they can see it.
Okay. So thank you to Asia for her wonderful presentation just now. Thank you, Dr. Greer, for that lovely introduction, and everyone else for joining us today. So I'm excited to talk about my project, which involves, or it's entitled, the Comprehensive Analysis for the Faster Diagnosis and Treatment of Leukemias. So we'll see if I can get this overview slide to come up here. So essentially, my quick overview for today is I'm briefly going to touch on cancer and our blood system. Is it coming, Asia? Okay, I'll try again. I just don't want it to skip ahead too far.
Sometimes you might have to use the arrow the first time.
Like the mouse? Oh,
Yeah, and then it should be able to work. There we go. It can work now.
Okay. All right, so for a brief overview today, I'll go over cancer and our blood system, blood cell differentiation that occurs in leukemia, and also some driver complexity, briefly touch on that, and what our current laboratory diagnosis pathways are, which leads directly into my hypothesis and the objectives, and a brief touch on the experimental approach that I'm taking to date, and some slides on preliminary data. So as I think all of us know, cancer is the uncontrolled growth of a population of cells. Now, when these cells grow in an uncontrolled fashion, the problem is that they disrupt homeostasis and normal bodily functions. This most often occurs through two main mechanisms, either a loss of function of a tumor suppressor gene or a gain of function of a proto-oncogene. Now, these driving events usually occur from a replication mistake that occurs in the DNA. And these replication mistakes provide that advantage to cell survival, resulting in the cancer itself. So what type of replication mistakes can occur is, I'm going to see if I can get this little cursor, laser pointer. Okay, so there's some different examples of replication mistakes. There's a single nucleotide variant, or SNV for short, which is when one particular nucleotide changes to be a different one than it's supposed to be. So in this example, there's a thymine here, and below there's a cytosine, or a C. There's also something called a multinucleotide variant, and that is when it's a collection of nucleotides in a row that are either duplicated, inserted, or deleted from the sequence. And this stretch of nucleotides is not quite as big as a full exon or a gene. So when it's a full exon sequence or a full gene, it can either be duplicated or deleted as well to be another type of driving event or another type of replication mistake. And also there is translocations. So that is when two segments of genes misalign, usually during mitosis, when the sister chromatids are aligning, and there can be two gene sequences that cross in the wrong fashion and join and make an abnormally fused product. So depending on the gene that these variant drivers occur in and the type of cell that they occur in, influences what type of cancer is produced. And most commonly, these driving events occur in cell functions that are involved in cell division, DNA replication, transcriptional activation. Sorry, I'm getting a little crazy with the cursor here, and double-stranded DNA break repair. So that's just an example on the bottom right-hand side of some functions that can be implicated in various types of cancers. Now, when we look at our blood system is responsible for producing millions of blood cells each day. And in the bone marrow is where our hematopoietic stem cells reside. And those stem cells have a job to first differentiate into either the myeloid pathway or the lymphoid pathway. So when they do this differentiation, the lymphoid pathway then further differentiates into lymphocytes on the right-hand side there that become our B cells, our T cells, and our natural killer cells. And on the left-hand side here, the myeloid pathway. Sorry, I'm really crazy with this cursor. Okay, so this myeloid stem cell here has the choice to differentiate into multiple different cells. And these cells go on to become either platelets, red blood cells, monocytes, granulocytes. Platelets obviously are involved in clotting in the peripheral blood system. Monocytes and granulocytes are involved in immune function and fighting off infection. So in a normal state, our bone marrow has those immature cells, so those immature blasts. And in our peripheral blood system, we have the mature cells circulating. But what happens in acute leukemia specifically is that we have the release of these immature stem cells or these immature blasts at various parts of the process that can then get released into the periphery, and that's what causes the acute leukemia. There's also other types of chronic blood cancers. So there are myeloproliferative neoplasms, and what that is a group of diseases that have an increase in proliferation of a specific cell type or potentially multiple cell types, depending on what the clinical phenotype is of that individual. But in essence, in this cube diagram here along the left, you can see that MPN results from that increase in proliferation. And it can also, with the loss of differentiation of those cells, evolve into a secondary AML or an acute myeloid leukemia. Myelodysplastic syndromes is another group of diseases that, contrary to MPN, where there's a lot of proliferation, in MDS, there's a lack or an absence of those cell types in the blood system. So there also is basically a dysfunctional maturation with respect to those cells. And with further added loss of differentiation, you can also evolve into a secondary AML. And then in between here is just showing that there can also be there's a lot of phenotypic overlap in these diseases, and there can be also MPN and MDS phenotypes together. So this is a slide just showing some of the major driving events of acute myeloid leukemia. So going back to those 6 cell functions that we talked about a bit earlier, there's some gene names listed here that are involved in epigenetic instability, or they're involved in chromatin packaging and remodeling. They're involved in transcription factors for turning genes on or off. They're also involved in double-strand break repair. So these genes here have been known to drive AML. And then similarly, when we look at this list, this is a list of another set of genes that are known to drive ALL, or acute lymphocytic leukemia. And these genes are also involved in kinase, so driving proliferation, or also transcription factors and some other aneuploidy events that can occur. So how do we currently diagnose a leukemia when an individual comes into the hospital? So when a patient comes in through the ER, they get many tests ordered. They get a routine CBC that's done. They have chemistry assessments that are done. There's a morphological assessment that can also be done, or that is done, on the bone marrow by the hematopathologist to look for any types of abnormalities that exist there. There's also flow cytometry that's done to look at any cell surface antigen markers that are expressed to see the lineage that's present. And there's also genetic assessment that is done on samples themselves. So on the right-hand side, when we're looking at the genetic testing that we do here, there's multiple different types of tests that we use in order to diagnose a leukemia or a myeloid neoplasm. So we have karyotyping, which we do to examine for aneuploidy and translocation events that are occurring that we can see at the chromosome level. We also do fluorescent in situ hybridization, which is to examine for copy number, amplification, or deletion events that occur that you can see at the cellular level. We have rapid standalone tests that we do with respect to, now these are for precise targeted variants that are well documented in the literature that we know have actionable targets. We have rapid standalone tests for that. And we also have them for some DNA variants and also for some RNA fusion products as well, like BCR-ABL and PML-RARA. We have smaller panels that operate as a standalone. We have one for MPN that looks at CALR, JAK2, MPL. So 3 genes that are involved in order to get information on any driving events that might be there, and also some prognostic information. And then we have a larger sequencing panel that we use, next generation panel, that includes targets that are the SNV and the MNV type. So driving events, sorry, that are SNV and MNV type. So if we have all those tests, what's the problem? Like, why are we here today? So we're here because we have all these standalone tests, and some of those are needed and some of those will absolutely continue. And the benefit of these standalone tests is that they're generally rapid, they're generally very precise, and like I said, they're targeted, they're associated with known targeted therapeutics. Now, the costs to these tests, though, is that they're limited in scope. They often involve repetitive strain because there are multiple smaller tests that we don't really have the capability of automating them as easily. And there's multiple analysis that has to get done in order to pull all that information together for that patient diagnosis. So if we look at a comprehensive sequencing approach at the bottom here, there's still going to be costs with respect to the equipment. We still have to have equipment. We still have to do analysis. We still have to store the data. But there's the benefit of being able to detect more driving events than we thought might have been there, do a more in-depth analysis, look at copy number changes, and even get into the possibility of risk stratification with respect to various driving events and phenotypes and clinical outcomes. So this ties into my hypothesis, which is that a targeted massively parallel sequencing assay with broader gene and DNA copy number coverage will enable us to detect multiple drivers that are involved in leukemia and myeloid neoplasm with increased efficiency and efficacy. So I aim to test this hypothesis through two main objectives. So on the left-hand side, my first objective is to design and clinically validate a comprehensive molecular profiling assay for multiple types of drivers. And on the right-hand side here, to develop technical and clinical standards for the interpretation and reporting of those detected variants. So essentially, we're doing assay development, assay validation, and assay implementation. Now, I've broken down this further into 4 stages. So stage 1 involves assay development. 2 and 3 together encompass assay validation. And stage 4 is the assay implementation. So we can't actually assess the efficacy and the efficiency until we have all of the results and data to do so at that point after we've implemented this assay. So looking at the panel design. So through the wonderful minds of Dr. Quest and Dr. Ra, we designed this panel with 49 fully sequenced genes, 42 hot pot. See, I want to go for hot pot. This is the problem. Okay, 42 hotspot genes and 11 chromosomes were chosen for the inclusion of extra backbone coverage. So on the far right-hand side, that's the chart there of the various chromosomes we added in. And those chromosomes were specifically chosen because of their involvement in both myeloid and lymphoid leukemia. So we made sure we chose as many targets as possible to include that are both myeloid lymphoid. We also included some hereditary hematological targets as well, because we're trying to look to the future. And just a sort of a note, this panel is actually bigger it has an additional 32 fully sequenced genes than our current panel, and an additional 19 of the hotspot genes than our current assay in use. So we got that panel design down. Now what's next? Now we have to figure out, well, what method are we going to employ? So there's 2 main sequencing methods that are in use in genetics overall. There's a third one on the horizon, but I'm not getting into that one right now. So the 2 I'm talking about here are amplicon-based sequencing, which is on the left-hand side, and probe-mediated pull-down. So amplicon-based sequencing is where we extract the DNA, we design a set of primers that are specific to the genes of interest that we want to interrogate. We amplify up that DNA with those primers. We then have various steps that occur to do the ligation of the indices and quantify those libraries. We pool them and put them onto a biometric chip. That chip then goes through, most often, semiconductor sequencing. And that's where the nucleotides are flown across one at a time. And as they're incorporated into the replicating DNA strand, a pH change is measured. So that pH change is recorded by that chip. And then that information is aligned to produce the variant calls. So this method it has some advantages, right? It's relatively fast. Usually it can be automated. It also doesn't require very much DNA input. You need very little. We need 20 nanograms to run this on the left-hand side, okay? That's it. So it has that advantage for sure. Now, what about this probe-mediated pull-down? So what this method is we take that extracted DNA, and instead of amplifying it, we chop it into pieces, presumably random pieces, okay? meaning they're all different sizes. And then what we do is we ligate indices. There's a couple cleanup steps that we go through, and we make sure that the library is now all the same size. And then it's clustered onto a glass flow cell in the sequencer. And this flow cell then undergoes sequencing by synthesis, which is a fancy way of saying the nucleotides are fluorescently labeled. And as the nucleotides are incorporated into the growing strand, there's a fluorescent color that's released and interpreted by the computer. And then that computer aligns that data to detect the driver. So the pros of probe-mediated pull-down are that it has much better base accuracy. It has usually much more robust depth that can be obtained. You can more reliably do copy number detection because you're not amplifying up at the beginning. And also you can do a pretty accurate measure of allele status. But what allele status means is if you have two driving events or two mutations that are within close enough proximity, so they have to be within the same length as your library, you can tell whether those two variants are on the same or different chromosome. It's super fun. So for this project, we decided to go ahead with that method. So we're using the probe-mediated pull-down. We are using sequencing by synthesis. And once we have the data coming off the sequencer here, we need to do some run QC as our first step. So what we did is we did 96 samples into 3 batches of 32 samples per batch. The sample distribution used is listed there below. And overall, there's 84 unique samples so far. 9 replicates have been done, and 3 samples were used for limit of blank and limit of detection. So after the run QC, there's going to be copy number analysis that's done and variant detection that's done for the analysis. And we're lucky in this sample set, for most of the samples, we have the ability to look at orthogonal data. So we will be able to align these results with FISH and with flow cytometry. So that's stage 1 and 2 completed. Now, going into parts of stage 3, which is the quality control assessment. This here is a visualization of library yield that we had obtained for the first 3 runs that were done in the order that the samples were processed. So in the order the samples were physically located in the run. Now, every sample in this graph did pass our minimum requirement. So our minimum requirement of yield was 100. So the line, sorry, again with my laser here, 100 is the minimum, 100 nanograms was the minimum going forward. Everything past that, but if you notice with your eye, you can kind of see how there is a large variance here at the beginning, or a large scattering. It kind of comes tighter in the middle and then it scatters again at the end. And I thought this was extremely fascinating because I had never actually lined the data up like this before in the past. But essentially what this shows is there's just more variance in amplification from the ends of the runs. Now, it's not surprising because this is a manual process that was done to see that. Going into the next slide is a visualization of, this is now uniformity, sorry, a measure of uniformity by fold 80 by % on target reads across the sample types. So this visualization was done to look if there was any technical outliers that might exist based on the disease state that the sample was from. So because we're looking at AML, CLL, MDS, MPN, some of the samples had MDS and MPN overlap. And then we have normal DNA samples in here as well. I wanted to be sure that there wasn't any one population that was showing a significant difference in coverage quality. Because if that was happening, we need to correct it now before we move forward. So the check marks represent healthy control DNA that we obtained, and they overlap pretty much with all the other disease states. So that's good to see that there isn't a significant difference between how the healthy controls are covered versus the disease population. The solid black square, so there's 3 samples that are a solid black square. Those are the synthetic DNA samples that we ran. Those all had very high % on target reads, which isn't surprising because they are actually synthetic cell lines that are specifically a blend of myeloid and lymphoid targets. So they're enriched essentially for those targets. And they also have an increase in fold 80 uniformity, which is also, I'll explain that on the next slide. Fold 80 uniformity is a measure that tells you as you increase from the value of 1, you need more sequencing to reach a desired average coverage. So the synthetic controls coming into the right-hand side there is within reason of what we would expect because they're not a full genomic complement. They're missing a lot of genes that are in a normal genome. They're just a synthetic subset. But this was also another visualization. So this is the exact same data points from the previous slide, except for by run number instead of by sample type, just to ensure that there isn't any one particular run that suffered a dropout in coverage. So that completes the quality control assessment. And the next steps are to go through the DNA analysis in more detail for the SNV, MNV, and CNV results to calculate the sensitivity, specificity, the limit of detection, and the limit of blank. And then moving into stage 4 will be the efficacy and the efficiency. So the measure of efficacy will be based on our increased ability to detect variants or drivers now that we were missing before, that we couldn't see from our old panel. And our measure of efficiency will be the amount of test resources saved, because now we're able to run one assay and detect copy number and those other MNV and SNV drivers. So this is a slide where I'm just looking at, here's where we came from in the 1800s and here's where we're going. So essentially, there's been quite an evolution of molecular diagnostics. Everyone in this room knows that over the last while. To the technical experts in the field who are in the audience, I realize that is not a picture of fish. I'm sorry, but I couldn't find a better picture quickly for the slide. But what this is trying to show is the evolution of when tests came on board, when they were developed. And all of these tests have been able to come to the clinical lab because of valuable research that's done in universities across the world. And around 2000 is when we were able to start using next generation sequencing. It was a bit after that in reality in the clinical lab, but that's when we all know when the genome was first sequenced. But in order to get to full genomes, that might be the question on people's mind is, well, if you're developing this other panel again, why aren't you just going right to genomes? And it's a valid question and a valid point. And the problem is we can't jump there just yet until we have enough clinical frameworks in place and variant interpretation guidelines to understand the data that we're getting the sequencers. Because with all of these genes, there is an incredible amount of data. We need to actually understand the meaningful impact of that data before we jump, clinically speaking, into genomes for everyone. But that is going to be the future, no doubt. So I'd like to thank my supervisors, Dr. Graham Quest and Dr. Michael Raw, also the members of my supervisory committee, so Dr. Janine Diba, Dr. Catherine Trishkin, and Queen's faculty and staff, Dr. Peter Greer, my Raw lab colleagues, and my clinical genetics lab colleagues as well. Thank you everyone for your attention. And I will now take any questions anyone has.
Okay. Questions for Laura? Ron, go ahead.
Thank you for the talk. I have a question about the general perspective on say, for example, whole genome sequencing versus sequencing of particular panels or doing individual assays.
Yeah.
To what extent is the cost a factor? Because I think the number that's on the front is whole genome sequencing this one is something like $100, which on a clinical scale is probably not that much.
Absolutely, yeah.
So
$100, sign me up.
Yeah.
But then you have to interpret all that data.
Yeah.
And
that's
store all
how
that data.
How do like panels of assays and individual assays, how do they?
Yes, absolutely. So the question is with respect to whole genome sequencing isn't really that expensive with respect to running it technically, $100, let's do it. But why aren't we doing that on a resource level for the clinical?
Just to get an idea of what
For the cost, you want me to tell
How
you? the
much
cost?
is one of the
Yeah.
other, like a
Yeah.
test for BRC or?
Oh, so, well, we receive funding from the government for certain tests. So we receive $750 for a solid tumor NGS interrogation. So breast cancer that we were just talking about earlier is $750 to run that test. Fish, I believe, three. Susan, do you remember fish? 300 and something, I think, for fish. Yeah. But yes, they're under $1,000 usually for most of the cost of the reagents. But there's also a lot of validation time, tech time with respect to keeping all of these assays up to date. You have to do quality control, you have to calibrate all your machines. And really just the gap to whole genome at the moment is really the cost associated with all of that. So we have the technology, that is not the problem. It's the cost associated with storing all the data, interpreting all the data, and retrieval of the data, quite frankly, because a lot of the reason why we think whole genomes is better is you do it once, right? We do Peter's genomes tomorrow, his genome's not going to change. Great, we've got it done. But then we have to store it so that we can always access it, right? So that's the storage is really the problem. Yeah, and the expense.
But go ahead, finish your question. Well, I was gonna say that, it sounds like, the technical cost of generating the data is quite small.
Yes.
But what about, all of the annotation and all the analysis?
Yes, that is where the resources are.
Okay, so if you add
needed.
that on to the $100 or so, what is it
Well,
what is it
it's-
really cost?
So to do a whole genome right now is, again, we're not doing it clinically, we're just running it. It's not $100 in reality with respect to, they've had competitions where it can be $100, but it's not $100 in reality. It's around $1,000 for all the reagents.
Okay.
I can't really quantify all the hours of time it takes for people to do the interpretation.
Yeah, well, that's where I'm going with this, because there's
Yeah.
probably people in this room that have sat down with, I don't know if you've had your own genome sequence, they probably sent you a disc or something, and people go, here's my genome, but they have no idea what to do with them. So I'm guessing that there are some people in the room that have some skill sets on how to analyze a large data set like that. But that's gonna cost money,
Yes,
time, money.
yes, for sure. Yes, Marco.
When you're running like whole genome versus like a panel at scale, would you have to take sequencing depth into account? Like, is there any cost saving there as well?
Yes, so the question is, when you're running whole genome versus a targeted panel, is there a depth consideration for the coverage? So absolutely. The reason why people, and I mean vendors, will quote you $100 for genome is because they're doing about 20 times coverage across the board, which yes, as you go deeper in the sequence, so as you cover each nucleotide position more and more, it costs more in sequencing reagents and also data storage because the files are bigger, but mostly the reagents. So right now, because we need copy number coverage, we need about 200 coverage. That's the lowest we can go. We can't go down to 20 for somatic. Now there's other studies that are showing you can go down as low as 75 if you're doing germline and still get copy number from that. But what we're speaking to here is a somatic type of detection. So we need to make sure we have enough coverage to do that. Yeah, but you're right. You can save money by not doing as much reads or having a bigger batch.
So, Dr. Mueller has a question. Go ahead, Chris.
Hey, great talk. Really enjoyed it. Can you just talk about allele
Are
frequencies?
you there, Chris?
Can you hear me?
I can't hear you.
I was gonna say, do we have a microphone to hear or a
Well,
speaker?
no, Chris, you're muted. Can you?
Can you hear me now?
There
Can
you is. Go ahead, Chris.
you hear me now?
I think it might be us. That's the problem. Yeah.
Yeah, Chris, I can hear you, so they can't hear you in the audience.
Okay, hang on.
Sorry.
Okay.
Oh, there.
Sorry.
Sorry, Dr. Mueller. Can you hear us? Or can we hear you now? Can you?
I can try. Can you hear me?
We can hear you now.
Okay, great. So really enjoyed the talk. Can you just describe, the effective mutational allele frequency in your assay? Because obviously these aren't pure tumors you're looking at, right? They're mixed population.
Absolutely. So did everyone hear that question? Okay, so because we're dealing with leukemia and myeloid neoplasms, we're not dealing with a solid tumor. Our sample is always diluted out with a normal population of cells. So at the moment, with respect to the samples that we're using, they've all met the criteria to be, generally speaking, greater than 12% blasts. So when they're acute leukemia, so there's at least 20% of the cells in the periphery are those drivers of the cancer, in theory. Some of them go down as low as 10% blasts. And for the purposes of this part of the project, we are simply we're going to have a list of all the blast percentages and all of our driver detection and variant allele frequencies that we see, and see how low we can go with respect to the limit of detection there. There's another idea in the works that we are going to look at doing cell selection first. So actually cell selecting out the myeloid and lymphoid cells before we do the DNA extraction or the RNA extraction, so that we have an enrichment of the DNA and the RNA that could be containing those variant drivers. But we're not at that stage yet.
And so what about sequencing depth? Are you going to change that depending on that assessment?
So I'm not sure yet. Yeah. We probably will keep the depth the same just for consistency's sake, and see how much better our resolution is with the cell selection. And then, if I'm being fully transparent, we're probably going to go to a whole different assay and evolve it even more. And at that point, it'll be a different assay. So we'll be looking at depth in a different way.
Thank you.
Yeah, thank you.
Gabby, go ahead.
Yeah.
I don't, it focuses on specific economic regions, and it does deep sequence of those regions.
Yes.
In terms of limitations of it, is there a possibility that SMBs or variants can be missed and focusing on specific regions of it? How do you discern which regions?
Yes,
Can you-
absolutely. So for amplicon-based sequencing, Gabby's question is, what limitations does amplicon-based sequencing have based on the fact that it's using primer design as its first step? So absolutely, you are only going to find what you're looking for, number one. So you're only going to find genes in areas that you're amplifying. And even further to that, with amplicon-based sequencing, there is a lot of artifacts that can occur. So false positives, actually, that can occur right on the edges of the amplicons that are made. So that's also another problem. The other problems aren't necessarily to do with amplicon-based sequencing itself, but it's more the semiconductor sequencing. So because semiconductor sequencing relies on measuring pH change, it has problems with multiple homopolymer regions. So where there's multiple nucleotides in a row, and when I say multiple, I mean 3. As soon as there is 3 Gs, 3 Cs, 3 As, or 3 Ts, there's a problem. It can't find if there's a duplication or a deletion. And we actually had that problem, saw that problem, proved that problem to be true in the clinical lab. And that's why we needed to move. In 2018, we had to move our cardiac assay to probe-mediated pull-down for that exact reason. So usually my recommendation would be, again, amplicon-based sequencing isn't horrible. There's definitely a purpose for it. But you need to very carefully look at what your genes are that you're interrogating, what's your purpose, what's your disease state of what you're testing, in order to decide which method is the best one to employ, for sure.
Quickly, because we're running out of time. But
Yeah.
just 2 comments. One is on one of your methodology slides.
Yes.
You got a picture of a zebrafish, which is a bit confusing.
Yeah.
So, I mean, you're not doing this on fish.
No.
You know what I mean?
Yeah, I know. I just took, you mean for the fish microscopy one?
No. Not fish microscopy. Here, sample acquisition. You got a fish.
Oh, yeah. That's just because,
I mean, so that, yeah. I mean, anyway.
yeah.
He couldn't listen after that.
See, that was a test and you won. David, you won.
Anyways, everybody. That was not what I really.
That,
wanted.
yeah,
So, humans, not fish.
yes, correct.
Let's
I'm,
fish later.
we'll do fish later. I'm sure they get leukemia.
Preliminary data, slide number 3.
Yes. Preliminary data, slide number 3, yes.
This.
Oh, the first one?
This one here, this is good.
Okay.
I don't know.
Oh gosh, it's still
So
going,
this is a
sorry.
This is quality assurance. And this uniformity number, the hell is that? And influenced by pseudogenes and copy number variations. Anyway, maybe you could explain.
Yes, so whole data uniformity is a new measure that's in the field. It started recently. It is a little bit arbitrary, yes, because it's basically giving an estimation that as the number gets higher from 1, you need more, your uniformity goes down. So you need more sequencing coverage.
Need more depth of
You need more depth.
Okay.
Yes, exactly.
But what does the ED mean? I meant to ask you this the other day. What is that? when you
Oh,
say
it's
whole
because of
80%?
80%. So it's measuring the coverage at each base across the samples that are in the calculation to as a metric of 80%, reaching 80% coverage to the mean. So that'll be influenced by pseudogenes in the sense that if you have a lot of genes in your targeted panel that also have pseudogenes and your mapping is misaligning, you're going to lose, because this is a measure only on the on-target reads, not on all the raw reads.
And the copy number variance would increase, you get many more reads from the copy. The increase
Correct.
in numbers of copies.
Correct. But you'll get, so what happens is you'll get, if you have an abnormality in a sample where there is that copy number, yeah, you'll get a lot more reads going into that. And it will actually lessen some of the reads in other areas because there's only so much coverage that's happening across that sample. And so you'll have a drop in so-called uniformity simply because of that abnormality that's present.
Thank
Yeah. Thank you.
Monty, go ahead.
Very quick answer the question. Why
Yeah.
is no one doing this? Why is no one thinking this is kind of panel?
Yeah, it's because honestly, in clinical genetics, we have to kind of take steps, right? Because we have to always be mindful that all of the resources that are used for this is public money, right? It's our healthcare dollars. So we can't just go big and bold all the time right away. We have to have the tests be meaningful and be not as resource level appropriate. So yes, we have those standalone tests that will exist and they will always exist because we do need rapid results for certain reasons. But whenever you get into expanding into larger data sets, we take incremental steps in improving the technology and adopting it. Yes, yeah.
Okay. Thanks very much.
All right, I'll have a